Okay, YouTube is online.
Okay, great. All right, thank you so much. I'll get started.
Yeah.
All right. Stops here. Okay, cool. All right. Back for another next day of streaming of the Exomeo Fragmissions project. Just do something like that. Okay. Yeah, there we go. Okay, so let's see here. Today, we're going to spend a little bit more time looking at the genetic variants, and then also looking at some of the radio stuff, because I didn't really get to go through that much yesterday. Some of the radio aspects of the project and talking over that. But yeah, it's mainly going to be a work day as I sort of try and understand a bit more about these variants, genetic variants like I was going through yesterday. Start maybe exploring some of the ways to mutate this with code, and perhaps also working a little bit on the code for the radio part of things. So I've been talking a lot about this radio thing. This project really has two parts. One, looking at these ideas of genetic variants and sort of the ways in which there's this research now on sort of the possible genetic basis of being transgender, and sort of exploring this conceptually through concerns or thoughts about genetic determinism, or the ways in which different types of elements may come together to sort of lead to different types of identities, and sort of what are some of the material bases of that. But then I was also wanting to take some of this information and spread it, throughout sort of the radiophonic space. Different types of marginalized identities, such as being transgender, but there's, obviously many, many others, are marginalized. And one of the things that I wanted to do was to take some of this genetic data, some of this thing that's maybe overly deterministic, do some kinds of different types of data mutations with it, and then think about transmitting it within sort of a shortwave radio context in order to sort of blanket the globe, because shortwave radio is something that really it allows you to transmit information from one end of the globe to the other by bouncing signals off of the ionosphere and sort of infusing the radiophonic space of the Earth with these signals. And it's something that I've been fascinated with for a long time, because it fundamentally doesn't require a lot of technology. It doesn't require a lot of infrastructure. You basically need a radio, which can transmit and receive, and an antenna. And that's it. It's not like the internet where you need your computer and then the whole network that's behind it of all the servers, all the intermediaries, all the routers, all the undersea cables, satellite links, and so forth. With radio, you just fundamentally need the radio and an antenna, nothing else in between, except for the Earth, right? And for me, that's quite fascinating, sort of this relationship between the radio, the transmission, the data, the signals, the desires that are sent through those signals, and then the Earth, sort of this tight connection, and this relationship that's formed between all of these elements and all of these materials. And to me, it also speaks to this possibility of a DIY communication, which I think is very much in line with these ideas that we're exploring this week in Lab Kill Lab. And so, you know, that was one of the reasons why I wanted to incorporate this into the project, as a way to sort of have one particular, somewhat ephemeral output of this output of this project. So let me just pull up a little bit of how this would be working later in the week. So the types of frequencies that I'm going to be using to transmit require a ham radio license, an amateur radio license, which I have. My call sign is KC1EKK. And so this allows me the legal right to transmit, because you know, these frequencies are sort of controlled and demarcated in terms of what can happen on these frequencies at particular frequencies. And so particular bands, we call them, are allocated to amateur radio transmissions and experimentation. And so this is something that I find, you know, again, really quite amazing, that there are still these bands that are legally allowed, legally designated for this type of experimentation, which frankly is becoming a lot more difficult, I think, on networks such as the internet, with different types of walled gardens that exist these days with different platforms. Now, with that said, obviously there's all sorts of things that you can do with pirate radio and so on and so forth, which is a whole other topic and a whole other sort of can of worms to get into. But we're going to be doing this using sort of my ham radio license, which allows me to do this without the threat of the law coming down on me. And so I'm going to be doing this from Ljubljana with an omnidirectional antenna, and I'm going to be trying to do it with what's called QRP, which is low power shortwave radio transmission. Low power meaning about 5 watts or more. And we're going to be using a digital mode called FT8. Now, in ham radio, amateur radio, you can certainly transmit your voice. You can transmit Morse code. There's lots of people who do that a lot. For me, I'm going to be using this digital mode called FT8, which is designed to transmit short messages without a lot of power in very noisy environments. And so I'm going to show you a little bit of what that looks like in a moment. So that's sort of the radio equipment that I have here. Then at C-Lab, as part of Lab Kill Lab, there's a whole sort of radio setup. On the balcony, there's what's called a mini whip antenna, which is a very, very small, active, powered antenna, which is designed to work pretty efficiently over shortwave frequencies. There is additionally, so that is connected to a Raspberry Pi 4. Or yeah, let me talk through a little bit more. There's so the cable from the antenna then is connected to what's called a bias T, which actually provides the power for the antenna over the coaxial cable that connects to the antenna. That bias T is connected to what's called a ham it up, which allows for shortwave radio reception. And then that ham it up is connected to what's called an SDR dongle. SDR stands for software defined radio. And basically over the last decade or so, some people discovered that this particular chip that was designed to pick up television signals, digital television signals, could be modified to pick up all sorts of different types of radio signals. And so there's been a huge explosion of what's called software-defined radio, which basically uses that chip to pick up these signals and to do it all in software on the computer. So no longer do you really need expensive radio equipment with all sorts of analog components, which makes this, which used to make it extremely, extremely expensive. All you need is this SDR dongle, which plugs into your USB port on your computer, and that then, allows you to, basically pick up these signals just with an antenna connected to your laptop. So, and then this is all connected to a Raspberry Pi 4, which runs this software called OpenWebRx, which I will show you in a moment, that basically works with the software-defined radio and some other programs on the Raspberry Pi to allow you to scan this the shortwave radio frequencies, and then also decode some of these signals, such as FT8, like I was talking to you before, these digital modes. And then also in the C Lab space is sort of a computer and monitor, which connects to the Raspberry 4, that allows you to explore the OpenWebRx software, which I did not write. I just want to make clear that it's an open source software that I'm going to be doing some modifications of maybe later today or next week, or later in the week, I should say. And then, of course, the monitor that allows me to stream this into the space. So let me just set up a couple of things here. All right, I'm going to go to my desktop setup here. Okay, great. And then show you what this interface looks like. So I'm going to log in to the computer that is displaying the information from OpenWebRx in the C Lab space. So this is actually the live feed running very, very slowly. And I have no idea how it's going to look on YouTube, but you will just sort of have to take it for what it is. This interface gives you what's called a waterfall, which shows you the intensity of a signal, which is given in the colors over time. And so in this case, right now, I'm looking at what's called the 20-meter band, or around 14 megahertz. And a particular part of this band that's allocated informally for FT8, that digital mode I was telling you about. And so these signals that are coming down here are sort of zoomed in over here on this box on the left. So I'm just looking at a very, very small subset of the band here on the left. And so these signals come through here and then can be decoded into these messages down here. And these particular messages are the decodings of these signals here. And to most eyes, these messages look rather cryptic, probably, and quite meaningless, perhaps. But for those who work on amateur radio, you can probably recognize this as call signs. So everyone who transmits on amateur radio needs a call sign. Like I said, mine is KC1EKK. And so, yeah, these are the call signs that are transmitting right now, and are being decoded here. And so there's not a lot of space for signals here in terms of what you can transmit. And for the most part, it's rather boring. The types of things that are transmitted, call signs, what's called a grid square location to indicate where the transmitter is coming from, and then some information about the quality of the signal, like this -24, And then sort of a goodbye message here, R73, which basically stands for reception received. And 73 is sort of a standard thing for amateur radio people to say, kind regards, best regards, goodbye, etc. Okay, so we're going to be sort of piggybacking and fragmenting on this later in the week. Okay, so I think I will quit it from here. Well, one thing you can also see is over here, this type of signal, which has a sort of very intense red band in the middle of it and some sort of wide-ish other signals coming off of it, is a telltale indicator that this is a voice transmission. And this is probably some sort of shortwave radio station that is transmitting right now at just a little bit below 14 megahertz. And I should also say that these FT8 signals basically sound like dissonant tones, sort of warbling back and forth. That's just a characteristic of the transmissions, but I find it kind of interesting as well. And sort of in a longer term version of this project, I'd like to be maybe manipulating these modes in order to sort of highlight these kinds of sounds. Okay, so I think I'll finish up talking about there right now. One other thing that I wanted to say, and maybe I will go back to the webcam screen for that, is that I have a Telegram group that I've created. Oops, not Telegram, t.net, it's t.me. I have a Telegram group that I created called XMEO Fragmissions, where if you want to join, we can talk about some of these other things with the project and what we're going to, some of the conceptual ideas of the project and so forth. So let me also just edit something here. Let me just change this. Oops. Title. Okay. Yeah, that's something that I can't really edit. No, I can't. Okay. Let me just edit this. Okay, great. Cool. So let's see here. Let's maybe go over a little bit of what we were doing yesterday, and then jump into today's work. So yesterday, I was working on the website NoMeD at the end to look at these different types of variants that were in this paper. And I was trying to look for these variants on the NoMeD website to sort of get an idea of whether there is any sort of maybe trace of these particular variants that are associated with transgender people in this much larger database, which has like 120,000 people in it. Yeah, that's what I was doing yesterday. So I just need to take a look at something here. Cool. So, yeah, let's get back into this. And at some point, I may stop my monologue with this and just do some work on the computer. So we'll sort of take a look at that. Oh, okay. Let me go back to showing you what we were doing here on day one. We were also sort of exploring this VCF file format, which is used to convey information about variants. And we were sort of discerning that file format, looking through these file formats in order to get a better idea of the data and what we might be able to do with it. So let me pull up the file format specification. So yeah, maybe this would be a good place to begin, because I think there's some interesting things to maybe play around with here in terms of some possible code poetry or something like that. So I think I'm just going to work a little bit on that right now. And if you want to sort of ask questions or anything, join the Telegram group. Maybe I will also put that in the ticker so that you know where to join it. And then, yeah, that's a way of interacting with me while I'm working. So let me write something here. Conversation on Telegram. Okay, let me see how that's looking. Okay, I think that will come up at some point. Okay, so I think I'm just going to mute my mic for a bit and do some work on this.
Adriana? Adriana, can you hear me?
Yeah, I can hear you.
Oh, okay. Yeah, because I think it's just mute.
Yeah.
Now we can hear you.
Yeah. No, I was sort of saying on stream that I was just going to be working a little bit on the code and sort of muting it while I was doing that.
Okay.
If that's okay.
你要问他用什么 resolution 啊？你要问他用什么 resolution， 不然我们就完全没办法支撑，完全没办法看到。
等我一下。
不要那么强。
Is there some sort of problem? All right. Okay. I just need to step away for a little bit because I actually need to run to the bathroom. So I'll be right back. Right, and back. Okay, so let's maybe think a little bit here about what we're going to be doing. So I really sort of wanted to play around a bit with this format, and do some sort of code poetry with it. So let's think about the different things that we could potentially do with that. Okay. Let's see here. Just start some notes. Okay, so I'm just writing things in a notebook here. It'll be easier for me to do this, and then I'll sort of tape it up in my digital notes, which I'll then post to GitLab. Okay, so So I guess one of the things that we're interested in here is how to take a sort of technical file format and mutate it in some ways, but still sort of working within that format, mutate it to create some form of code poetry that we could then potentially encode in a particular way to do this data transmission that we're going to try later this week. So for this sort of thing, we can sort of take this format and sort of explode it in all sorts of ways. Excuse me. So we've got these info fields, which seem to have an ID that is just 2 characters, and then sort of a different types of data types, so integers, floats, strings, and so forth. And I guess some flags. Yeah, so DB is just a flag. Cool. Okay. And then the descriptions of these, and then those are given in the info field. Okay, so what sorts of things might we be interested in? So I started work here thinking about number of transient people in the world. So let's just maybe start writing some things down. Just a second here, because it says that ancestral allele is a string. Can that string be anything? Let me see here. String, no white space or semicolons permitted. Okay, so that may be kind of interesting. So let's maybe open up a new set of notes here. What did I call it yesterday? Oh no, it's right. Come on. Program. Sometimes Vim is super slow. For some reason, it should be quite fast. Okay. Day 001. So let's start day 002. Day 002. Let's create some notes. Okay, so let's say here in VCF format, strings can't have white space, commas, or semicolons. Okay. So then, okay, so we were thinking about number of trans people in the samples that we're looking at, potentially. Of course, number of transgender people in the world. Samples, number of people in world. Let me just take a look at something here. Buzz. Yeah, just a moment here. I'm going to mute my microphone. Okay, I'm just going to try and restart OBS and log back in order to change my streaming resolution. So I'll be right back.
Is on.
Okay, so I'm back, but yeah, still probably have pretty poor quality streaming. Let me just mute my mic for a moment.
嗨，泽毅。
嗨。
OK。他现在应该还在瞧网络的问题。
没关系。
那个来了，就是另外一位。Andrea 现在是不在，是不是？
他设定一下他的电脑。
他设定一下，因为他现在看起来没有在这里。
好。
说他，因为他不好太差了，所以请他就是过程中还会把自己屏幕都录下来，然后事后再上传上看。对。
但是我们现在只能等他，对不对？
对，他应该知道他。他在。
阿姨气氛。他什么背景感觉厉害。
Is on.
Feedback. Hello.
Hi.
The one and then Anna. 你听不到人家讲话。
这边有吗？
所以你有听到 Andrea 说话吗？
没有
对，我这边也没有。
你直接讲，跟你讲他。
Andrea, we cannot hear you.
We can hear you. 应该是那个啦。
Andrea 好像很小声他在讲话。
听不太到。
We can't hear you. Okay, Telegram.
Maybe you can change your audio device. No, we can 他重加。
这个东西最好都不要上 FB。
对啊，我都没有上。
其实就上最重要就好。
对，没有，现在只有
唐头的。
其他都没有。刚刚太多了。
Is on.
Okay, now I think this should be working again. Is it working? Or are we getting lots of feedback?
Yeah, it's okay, but may noisy.
Okay, yeah. So I think I know why that's going on. I have some complicated audio things going on here in order to do the live streaming. So let me disconnect one more time, and then I'll come back, and I think we'll be able to make this work.
Okay.
Okay, so hopefully this will work and there won't be a lot of feedback or anything like that.
Guys.
great. So, thank you for joining us, Dr. Wang. I really appreciate it. Sorry that the emails came so spread out in time.
Yeah, that's okay.
I don't know what it's like for you, but for me, Corona times, things have become. I've had a lot more work to do. It's become a lot more difficult. So yeah, I mean, I guess I wanted to talk to you a little bit about these ideas of genetic variation and sort of how they are studied and sort of like, I mean, I'm very interested in sort of how one even defines sort of variants and their relationship to reference genomes, and then what does it mean to be searching through these databases for variants that are found in other papers and so forth. So, maybe because from what I understand, you have a background in bioinformatics, so could you talk a little bit more about that and maybe what research you do?
A background in bioinformatics, but
Oops. Oh no, I lost him.
research. Yeah. So the major part of my work is try to develop some novel statistical method and to do gene and disease association studies. So we want to more focus on explore the genetic variant that might be related or connected or associated with some disease. And most question is about the GWAS study. That's mean what the genome-wide association study. We want to find out which genes or which group of genes related to some disease. Besides disease gene association studies, some people will focus on phenotype and genotype association study. Such phenotype not only disease, sometimes maybe the species or just like recently, we are more focused on whether there is some gene that we can call is Taiwanese gene that's only specified for Taiwanese and distinct or different from Chinese Han people. So we can define which phenotype we want to know and which genotype we want to find the relation between phenotype and genotype. Yeah.
So because I'm really sort of curious about this because I think it ties a lot into trying to understand this study that I've been working on or looking at. So can you talk a little bit more about how you do this statistical analysis and sort of how when you're searching through a whole genome, so you're searching through the whole genome, not the whole exome, the whole genome. How do you do this statistical analysis in order to find these variants as you're trying to search for?
Sometimes we will consider two types of such GWAS studies. One is the case control study, and the other is only case study. That means that we may have some case of interest. Maybe all these cases are transgender, and the control sample will include non-transgender individuals, and then we collect the genetic variant profile, and we can do sequencing directly collected the DNA to do sequencing, and then we can compare all the genes with the reference genomes and find out which variants can be represent the genetic profile of each individuals. And then we compare these two groups and find out the difference between two groups. And the difference of the different genetic variants, we will consider them they are candidate genes to explain why this group are transgender, or this group have diabetes 1, or this group have some special phenotype and the other not. Yeah. So you can we will call it a case control comparison study.
And so when I've been looking at the sort of things, right, this idea the reference genome comes up. What is this reference genome?
Sorry, I don't hear.
How is it made, especially given that there's such variation in the genome?
Sorry, I don't hear you.
Oh, that's okay. Let me maybe put the mic even closer here. So I was asking about the reference genome and how the reference genome is created. Are you able to hear me?
What's wrong with the?
It looks like it's working on my end. I'm just gonna post something in the chat here.
Can you hear me?
Are you able to hear me?
I don't hear you. Yeah.
Okay. I guess I can come back into Jitsi, but I think I should be able to be heard here. Is anyone from C-Lab still on? Can they hear me?
Yeah.
Okay. They can hear me at C-Lab, they said. So I don't know if something got muted on your end, Dr. Wang.
Yeah, I try to reconnect again. Sorry, I don't hear.
Uh-oh, okay. Okay. Are created given that there is such variation in the genetic code.
Yeah, I think the story should be back to 2 decades ago. There a woman we call is Taylor, Madame Taylor, and she was the first one, the first human being have DNA be sequenced by many scientists, and so the first human genome project was developed in 1986, and during that time, many scientists tried to sequencing a single human genome and belong to Madame Taylor. And so the result of Madame Taylor's genomes become reference sequencing for all the human beings around the world. So right now, we have different versions of this human genome project. The result of this human genome project. Right now, the version should be 39 or 38. Yeah, I think 39. Yeah, 39. 13 version of 39. And right now, when we do sequencing of just like you or me, when we do sequencing of our cell DNA, and we will compare the result of the sequencing of our DNA, CGAT or ATGC, and compare it to Madame Taylor's genomes, and find out which part is different. Because the DNA of most humans are the same. More than 99% are the same. So actually, the difference between different individuals' genome only less than maybe 0.01%. That means that we can create a relatively small profile to represent each individual's genome. And others, we can consider that all the other sequencing not be recorded. That means that all the human beings are the same at that part. So we will do variant coding. That means that you can use some software. That software, basically most software, they have Madame Taylor's genome be recorded in that database, and the software will directly compare your input file with the reference genome and create another profile. We call it variant profile, and such tool we call it variant coding. Yeah.
So I'm just curious as well, because there's these different versions, right? Which makes me think about like software versions, right? So what changes between these versions, saying from 37 to 38 to 39?
Yeah, you can because the sequence technology improve, then we can get more accurate profile of human genomes. And every 2 or 3 years, Human Genome Project, the group of Human Genome Project, they will publish or deliver a new version of a new correlation of the human genome data. Yeah. And actually, right now, in my opinion, I don't really trust the result of sequencing. The error rate is so high that I believe it is more than 2 or 3%. And based on which chromosome we're sequencing. For Y chromosome, most sequencing result of Y chromosome, we can consider they are not totally wrong, but maybe at least 40% of the sequence results are not accurate. Yeah, because the Y chromosome is very hard very difficult. It's very difficult to sequence in Y chromosome. Yeah.
And why is that? Like, what makes it so difficult?
Based on the sequencing technologies, there are two kinds of sequencing technology. The ancient type of sequencing technology is each step we only sequencing one single bit of DNA nucleotide. And right now, we call it parallel sequencing. That means that we digest all the DNA chromosome into many small fragments. And we can parallelly sequencing all these fragments together simultaneously at the same time. And then we try to, just like we just like we have to we call it we should connect all these fragments back to the original orders. but it's very difficult because there are many repeat small fragment repeat many times, and when the fragment relative to some such variance sequencing, we don't know how to connect them and find the right order of the sequence fragment. Yeah.
So, because that's one of the other things that I've been discovering in the research that I've been doing, what you're talking about, this noisiness of the sequencing, right? Because I've been trying to sort of understand exactly what that is and how it arises. So some of it you're saying is because of these repeated fragments. Are there other problems that lead to this error rate in the sequencing?
You mean the error rate?
Yeah. Because you were saying that you were saying that there was like an error rate of like 2 to 3%.
Yeah,
so I'm just
many,
curious. What other things lead to the errors for that, and sort of like how is that? Are the errors something that someone who is using this data knows about, right? Like, do you know when you have a sequence and you get the sequence back, do you get also an information about how much error there is in that sequence potentially?
Right now, when we do sequencing, we will check. We will based on the tutorial of the sequence machines, and to calculate quality control score or some error rate estimations. And right now, mostly we only accept the error, the possibility, we only consider the possible error rate less than 1%, and then we will accept the result of such sequencing. Yeah. In some regions, if the error rate is higher than 1%, we will do sequencing again, only focus on that fragment, or maybe we will just abandon it. Yeah, we just give up. Yeah. If we have some more resource, we will do the sequencing. If we don't have such resource, we will give up. Yeah.
Right. I mean, I find that sort of fascinating that there's parts of the genome that are so difficult to get information about, right? Is there anything that is functionally important about those regions that are hard to sequence, or are they mostly in what we think are sort of nonsense or useless sort of regions?
Yeah, what you talk about, maybe exon seq can be some kind of such sequencing. It's not sequencing all the DNA in human being, only sequencing the exon part. Yeah, we can consider that the exon part is more functional than intron part, and most proteins are coding in exon. So if we more focus on the functional protein, then we will more focus on exon part. The exon part is quite small compared to the whole genome data. For each individual, you can only less than 1% of whole human genome belong to exon part.
Really? Wow.
Yeah.
I did not know that.
Yeah. If we do for whole genome sequencing, right now we probably need to spend $1,000 US. But if we do exon seq, it's only cost maybe $100 US
Oh,
dollars.
wow.
Yeah,
Oh,
yeah, that's
wow.
quite cheap. Yeah,
Yeah,
yeah.
that is quite cheap.
But the
Yeah.
trick is that firstly, they have used some chemical to digest the chemical way to digest the DNA, and only fish out the exon fragment.
Mm. Mhm.
Right now, the accuracy of the fishing exon sequence is acceptable. So most technology or most genetic library will do exon-seq instead of whole genome sequencing.
Interesting. So, I'm wondering if we could talk a bit more about this research that you're doing in order to look at the genetic variants that might be associated with people in Taiwan. Are you looking at this within exomes? Are you looking at it whole genome? What are you finding? What does it mean potentially? Because I'm quite interested in that.
For me, during the last decade, I spent much of my time to try to discover some human histories
Mm.
by DNA research. That means that in Taiwan, many Taiwanese believe that, or many historical textbooks tell most of Taiwanese that we all belong to Han Chinese people. We came from Chinese, we came from mainland of China, and maybe 400 or 300 years ago. But if based on human genome data, it's really true that Taiwanese have no difference compared with Han Chinese human genome. But actually, we find out that Taiwanese have some unique signature, some unique feature that differs from Han Chinese. So based on DNA study, we try to understand why these people different from Han, but we've been told we are Han. Yeah, it's quite interesting. So such study we call it the molecular anthropology. Yeah,
Okay.
we use DNA and to trace the human migration history. And for me, I'm actually, in my opinion, right now I believe that there is no single group of Han Chinese based on DNA study. At least there should be Northern Han and Southern Han. And for Taiwan, for most of the Taiwanese, we are quite a little different from Southern Han Chinese people. Yeah.
So, because I guess I'm trying to understand this also in relationship to what I think I have read about in the past, but correct me if I'm wrong, about like using mitochondrial DNA to do some of this research as well, some of this kind of like historical and not really anthropological, but almost archaeological sort of understanding of migration patterns and so forth. Have you been able to sort of um, look at the relationships between the two, between sort of the genome and mitochondrial DNA and sort of any potential traces of that separate being from Taiwan, being from mainland China? Have you been able to do that within mitochondrial DNA?
Yeah. Mitochondrial DNA were used to trace we call it, maybe we can call it genetic blood from motherhood. Yeah, that means that we can trace the the mitochondria based on the sequencing result of mitochondria, and we try to know our mother's original, where they came from. Yeah. And when we compare the Northern Han, Taiwan Han, and Southern Han, these three groups, we find out that the Southern Han and Taiwan Han, their mitochondrial DNA can be traced back to Southeast Asia people. And for Northern Han and Southern Han and Taiwan Han, their Y chromosome DNA, that means that the blood from their father most can be traced back to Northeast Asia populations. And if we believe that the Han people or Taiwan Han people, the ancestor of all this group, and belong to two ancient population. One is Northeast Asia and the other is Southeast Asia. We were quite interested in finding out that the feature of mitochondria are quite obviously from the Southeast Asia. Yeah. So many people try to, based on DNA study or based on some archaeology study, to explore what happened maybe 10,000 or 20,000 years ago. What happened?
Right.
Yeah.
Right. Yeah. I mean, I find that really interesting, having this trace of these histories within this material that, you know, maybe other types of traces were lost, you know, like writing or, you know, clothing or things like that, that it can also can remain within the DNA. I'm curious if there's, because I don't know this about Taiwan at all, but I'm curious about sort of prior indigenous groups there and different types of traces that you might be able to see about mixing between any indigenous groups in Taiwan and then sort of people from elsewhere who moved to the islands later.
You mean Australia?
No, not Australian. I'm saying maybe native people who were there before. People who, I don't know if this is the case about Taiwan. You know, if there were people already living on the island before. These studies that you're talking about are sort of finding information about the people who are already living on the island, or what? So I'm curious, you know, because like, you know, in the U.S., for example, you can sort of look at how you know, the Native Americans, right, through colonialization and everything, the mixing of that, right? And so I guess I'm just curious about that. You know, when did when are some of the earliest traces of people in Taiwan, and sort of like, is that sort of what you were talking about here in terms of finding these variants about Taiwan?
In human migration history, it's based on mitochondrial DNA study and Y chromosome studies. All the human beings can be traced to Africa because all the mitochondrial DNA, all the current people's mitochondrial DNA, we can trace them back to many ancient type, and that ancient type of mitochondrial DNA be preserved in current Africa populations. And for European people, we believe that they can be divided into three ancient groups. One is we call it Middle East farmer, and the other is Hungarians from the North Siberia, and these two parts, one is migrated from the Middle East to South Europe, and the hunting-gatherers group, they maybe travel from Middle East to Siberia, and they divided into the group. One is migrate to east, the other migrate to west. We can divide most of the European people into several groups, but their ancient feature can be traced back to one is North Europe and the other is Southern Europe. For North Europe, we can consider them as they are a combination of the hunting-gatherer and the Middle East farmer group, but the major part is hunting-gatherer group, and for Southern European, the major part is Middle East farmer group. And for other, just like Australia native people or Austronesian speaking people in Taiwan native people, they be traced back to the people they migrate through India and through South Asia and to the island Southeast Asia. And some of them migrate to New Zealand and Australia, and some migrate to Hawaii. Yeah.
Okay, interesting. Maybe because I also wanted to sort of get more into, I guess, some of the statistical analyses you're developing. I mean, I don't have my statistics are very. I have a background in the sciences, but it's been a very long time since I've been working in the sciences and statistics. But I'm curious if you could talk a little bit about how you do these sorts of studies in order to identify these variants. What sorts of analyses, what sort of statistics or calculations or comparisons are you doing to try and do this research that you do?
Right now, we have most GWAS associated study. Most researchers, when they do GWAS associated studies, we will follow. We can call it SOP. Okay. That means that based on the variant coding profile, or genetic variance profile, and first we will compare two groups, case group and control group, and find out if they can be considered same genetic admixture. That means that we want to make sure that these two groups have the same genetic background, just like they all belong to Caucasian, or they all belong to Chinese Han, or they all belong to Chinese Taiwan Han. And after that, after such genetic background structure studies, when we make sure that these two groups have the same genetic background, we will more focus on to find out which different variants can be relative to disease. Yeah. Because they have the similar genetic background, so the only genetic difference can be considered relative to their phenotype difference. Yeah. Mm-hmm. And we follow the tutorial of several GWAS software, and they can help us to do structural variance genetic background study, and then to find out which variance relative to disease based on statistical tests. And such statistical tests, the naive statistical tests, we can consider them as such as t-test. Just like we compare the frequency of specific genetic variant. If in normal people, normal control people, the frequency, the gene frequency of some specific variant is maybe 2% or 3%. But in case group, if we discover that the gene frequency is significantly higher than 2 or 3, or significantly lower than 2 or 3, we'll consider that such difference between gene frequency, we will consider they maybe can be related to the disease. Just like when we consider if you read some literature, they will tell you that we find out some specific genetic variants have higher frequency or more significant frequent in some specific group. Yeah. Mm-hmm. Just like for some people, they have some specific tumor or cancers. We find out that some specific genes have easy to find their variants in such group. Yeah.
Interesting. So I'm curious about this, you know, for the reasons that we were emailing about because of this study, right? Mm-hmm. And you know, so say I am looking in a database like GNOMAD or something like that has all of these exomes, and you know, I look, you know, I go to that original study, I find the variants that they found in this cohort of transgender people. I then go search for this variant within the GNOMAD database, and I find it. Maybe there's only one allele in the database of 120,000. I'm curious from sort of your sort of statistical analysis of these things and your statistical analysis of these studies, or thinking about these studies, what sort of probability could one say that if there's only one person who has this variant in the GNOMAD database and this variant was found only within this study, that person, unknown person in the GNOMAD database, may be transgender, let's say? What it's how, I mean, obviously that is not a scientific question to ask, but more of a conceptual question in a sense. Like, how possible is that? Let's say.
I think it's very difficult to based on the genetic variants to predict whether an individual is a transgender or not. That's why we call it association study and not cause effect relation study, because we cannot find the genetic cause that produce the phenotypic effect of transgender or some phenotype of interest. So all this relation is only show they have a statistical significantly difference. Yeah, that means that when we compare these two groups, the difference can meet the requirement of statistical significance. Yeah. And it's very tricky that just like most of GWAS study cannot be repeat. Some research group find out some genetic variants related to some specific phenotype, but all the other research group cannot repeat their finding. because sometimes it's only statistical significance, but they are not really cause and effect relationship.
It's interesting because back when because I trained as a scientist, I thought I was going to go into neuroscience. I spent a number of years working in a neuroimaging lab. So I was doing functional magnetic resonance imaging of the brain. And so we had a very similar problem in that, right? Because you scan the brain during a task, and it's very hard to do sort of a causation, right? Study. All you can do is say, well, we asked these people to do this task, and compared to the control, and compared to this sort of, way of, fragmenting the brain, these areas are more active than these other areas. And that's basically all you can say, right? And there's also been a similar problem about replicating these studies, right? There's a very famous sort of joke study that was done to illustrate this point. Basically, they put a dead salmon, the fish, in the scanner and found that parts of the dead fish's brain were active as a way to illustrate these statistical problems. Exactly. That you can get all of these sorts of false positives just by virtue of the statistics. So yeah, I'm sort of very familiar with these issues. But I'm curious. I don't know if you would be willing to speculate a little bit. What do these what are these variants that really? What are they? Like if if we see all this variation and we're not exactly sure whether it's causes any changes in the phenotype, why? Like what is like what does it do? What is its purpose? Is it just a trace of differences or what? I'm just curious about your speculations about this.
That's why some serious studies should be more focused on twin study.
Yes.
They have to make sure that if different individuals belong to same twin, the twin have different phenotype. One is transgender, the other is not. But actually, they have the same genetic background. They have all the same genetic variants. But why they different? Yeah, that's why when we do twin studies, we try to exclude, try to find out what will affect the phenotype, and not through the genetics vector, but through other vectors. Yeah. So that's why actually I'm in my opinion, I don't really can accept the genetic can explain everything. I think they only can explain 10 or 20% of phenotype.
Right. Yeah, no, totally. And I mean that's really interesting to sort of hear you say that, because obviously you and other scientists know about sort of the complexity of this, and that genetics does not determine everything. But I also feel like in culture or in sort of a public discourse about this, there's this belief, of almost a determinism of the genetics, right? Especially with, I don't know what it's like in Taiwan, but in the US, there's a lot of people doing these ancestry DNA studies, right? So they go, they send out their DNA, they say, okay, I'm 10%, West African and 90% Northern European or something like that, right? And so that then becomes this determination of who they are, right? And so, I'm really glad to hear, you talk about that in terms of, how little perhaps the genes actually contribute to the phenotypes. But I'm also curious here, because you're talking about the difficulty of doing this in humans. What about genetic variant studies in sort of other mammals or so forth? Has there been much done on that?
Yeah, I think they have the same problem as human beings. Some species maybe have more difficult problem when do their DNA research. Yeah, I think they have the same issue. Yeah, but for we call it maybe we can consider that besides human genome studies, other study of other species, they more curious about the evolution history or evolution relation between different species, and based on molecular evolution's history, they try to some people they try to recreate new species based on genetic technique, or just like most of microorganisms cannot be cultivated in lab, and at least 99% of microbials cannot be successfully be cultivated based on lab technique. But we still want to research the microbial or microbiome or all these microbial species, and based on DNA studies. There is the other way we call it metagenomics. That means that we collect all the DNA. They belong to many species in some specific environment, and we directly sequencing all the DNA, and then we have to we call it being, or try to divide it into different groups, or try to find out new species. Yeah.
Oh, that's.
But
okay.
The same problem is that the DNA technique cannot take us to the end. Yeah. We only can guess what happened. We only can guess, and we cannot make sure what really happens. Yeah, in such a tiny world.
I mean, that's also really fascinating to me, this idea of metagenomics. I had never heard of it, because for another project that I'm working on, I'm thinking about, say, the skin microbiome, and the different types of fungi and bacteria that live in the skin. And so that's an interesting thing to think about, sort of the sequencing and the potential genetic relationships between these organisms and one's body, perhaps. I mean, it sounds like almost there needs to be these studies of, like, a single mouse, with if you're doing mouse research in a lab, you have to have separate sequences for each mouse in order to sort of look at these variants. I mean, I find that really sort of interesting. I guess since we're almost done here, right? Yeah, I guess I'm just curious if there's anything else about your research on variants, or your research in general that you think it would be interesting for me to know, given what you know of my project?
I think about the if we about the transgender study, I will more curiously or try to want to know or want to maybe if I will do such study, I will more focus on to trace the gene history. I believe that in some ancient time, there should be some culture or there should be some genetic background and some environment conditions trigger that such genes they related to some brain functions and that related to transgender. Yeah. And if the acceptance of frequency of transgender in some specific human population higher than the other, and I believe it's not only genetic difference, there may be some cultural difference or maybe some environment condition difference trigger that some genetic factor can be preserved and evolved from ancient time to now to prevent to now and make such two differences have two different frequency of transgender individual. Yeah. So I will more interested to find out whether if there really exists some ancient population group, they have quite different culture compared to current human populations. Maybe they more sexual equality or more diversity in many social functions or in many social environment create that such ancient population has more diversity in human genetic and in phenotype and in human phenotype. Yeah.
I actually find that incredibly beautiful because it's so much of these types of behaviors or identities are lost to history in a sense, right?
Yeah.
Because we weren't able to write it down. There's no trace of it, but this idea that maybe the genetic code, while perhaps not determining things, also creates a history of this and sort of is our history. That I find really beautiful and powerful, is sort of both scientifically but also conceptually. So wow, that's something, you gave me something really to think about there, because because I've been sort of I did a very little amount of research earlier this year in terms of trying to look at questions of sexualities and identities and gender identities and so forth, going back into sort of prehistory, right? Mesolithic period and so forth, right? And of course, there's not a lot of traces of that because of time and things degrade. But if that is found in the genetic code, wow, that would be really interesting. That's super fascinating. I think maybe we should just end there because that's like a beautiful, it's a beautiful thought.
Okay.
So thank you, Dr. Wang, so much for taking the time and
thank you.
generosity and all of this. I learned so much talking to you, and you've given me a lot to think about here.
Yeah.
I wish I could have been there in Taiwan, but I'm glad to be able to at least do this remotely.
Okay. Okay. Thank
Okay,
you.
great. Thank you so
Okay.
much.
Good.
Okay,
Okay.
bye-bye.
Bye bye. Uh-huh.
Cool. Okay, I'm just going to disconnect and then reconnect here with OBS again and take a quick break to get myself some coffee and then maybe make some notes about this really fascinating conversation that may sort of take this project in some interesting directions. So let me just do that and let me reconnect. Excuse me, and I'll be right back.
On.
Okay, let me see here. Yeah, so I'm live here with video and everything. I'm just gonna get some coffee and then I'll come back. So the stream is still a bit on pause as I'm waiting for my coffee, need my coffee, but then I will be on hopefully within a couple of minutes.
Yes, she will be moderator.
And she will.
Adriana?
Yeah, I'm here. I'm here. I was just drinking some coffee.
And Yukiko is actually here.
Okay.
We want to talk to her, and she has a question for you.
Hello,
okay.
Adriana. Nice to meet you. I don't know if you can see us. Can you see me?
Yeah, I can see you. Yeah.
Yeah. So it's nice to meet you online. So it's pity that we don't have you here. But yeah, and today I was here, but there's some troubles on online the TT program, so I couldn't get exactly what all the things. But I have some fundamental questions because your work is still for me enigmatic. Sorry, but you've been sending and transmitting the data from Slovenia by ham radio, and it was easy to visualize on display.
Well, I will be doing that later. I will be doing that later in the week. But what is on display right now with the radio is other people who are transmitting using this data mode. I will be using that later in the week. So this is just sort of there right now to sort of illustrate, something about what we're going to be doing later in the week, right? And so then this at the beginning of the week is more the stream that you're seeing right now that's being projected and so forth. So working on the types of data and information that I will then be transmitting later in the week. Does that make sense?
Sorry, the sound is not so clear. Sorry to say.
Oh no.
So, yeah, can you explain a bit simply and maybe again? Sorry.
Yeah.
And I'm asking. This is a real-time data streaming and visualization, right? This is one question.
It is. Just one moment. I have some coffee on the stove that I need to take off.
Yeah. And it's already online. It's still online.
Yeah.
Because it's more internal question.
Yeah, but then I think we're not going formal.
So then, what kind of data exactly you are transmitting? Physical data of transgender or gender people, or?
Yeah. So I'm unmuted. I will be transmitting some types of poetic representations of genetic information supposedly about transgender people. So part of this project is about the difficulties of using genetics to define particular identities in people. So I had this very interesting conversation in the previous hour with Dr. Wang, and he was telling me this interesting thought that he had that possibly within the genetic code can find something about the histories of transgender people and so forth, because he was interested in a potential earlier type of society where being transgender or being gay or homosexual and so forth was more accepted, and thus that may have had a trace in the genetic code, which may be something related to these genetic variants, right? So what I'm going to be doing is looking at databases of human variation in their genetics, and then sort of pulling out parts of that that may be associated with being transgender, and then poetically recombining that with some of the data analysis tools and file formats that are used for genetic analysis. And then
Write the text on this Forking
Live streaming is on.
More.
okay,
Also,
yes, you can definitely email me, and I will send you more stuff. I mean, I think one of the great things about the way that Shuli has structured this week is that the projects are developing during the week, right?
Yeah. Okay.
So,
We will know more about that because
exactly.
it's in progress.
Exactly, it's in progress. And so already after this conversation with Dr. Wong this morning, I have some different ideas about where to go with this. So definitely send me an email, and I will, answer whatever questions you may have.
And how do you show this progress? Because maybe one day by day, you have a new information, some new source, and new result. How to say show this in a form of installation or somehow for the people, for the visitors,
Right.
around.
Right. So what I have done, I'm going to switch to a different setup here. Okay. So I have begun posting some of the notes and early codes that I'm editing on this code repository. So, for example, here I have all of the notes that I was taking yesterday. Let's see, a bit of time to load up here. Okay, so different notes, different variants that I'm searching for, and so forth. And so one of the things that I'm going to do is maybe in the next hour or so of the streaming is think of creating some visual representation of this for every day that can be printed out, because this was something that I was talking with Shuli about earlier. So I'm keeping a record of everything. We obviously have the streams that are posted to YouTube. And, something that could happen is that maybe the previous day's stream runs on a loop on the television, on the projections when I'm not transmitting, when I'm not streaming. Yeah, so those are some of the ideas that I've had.
Okay. Yeah. Thank you. Yeah.
Okay,
Nice to
thank
talk
you.
with you. Yeah. Arigato. Thank
Yes,
you.
nice to talk to you.
Yeah. Hi.
Hello.
Hi.
Hey.
So, can continue?
Yes, I will continue. I'm going to get back to my coffee, which almost flew over on the stove, and then I will continue.
Okay.
Okay, I'm back. Looks like everything is going. I've got some coffee. So, because I'm definitely not a morning person, even though I'm only starting at 9 a.m. here, which is really not that early. For me, it's early. So, I definitely need my coffee. Okay, so basically what I would like to do here is maybe think about that conversation that I just had with Dr. Wong, which, yeah, it was so beautiful. I was almost starting to cry at the end because, you know, one of the things about being transgender, let's say transito, being being other, which many of us are, of course, but being as part of a minority or a type of identity or something of this sort that is marginalized and has been marginalized a lot in society. Something about that, you know, we often don't have a lot of history. We often don't have a lot of records of our past because, you know, the majority didn't want that to exist. You know, it's the same thing with different types of Native American peoples, Aboriginal peoples, and so forth around the world. Many, many, many different types of people, of course. And so, you know, one of the activities of colonialism or of control is to sort of erase these histories. And, you know, there's a lot of discourse, at least within Western media these days, about being transgender, that it's some sort of new fad, right? There's a lot of concern by some people that people, you know, children who are trying to be transgender are just following a fad, right? Which is really sort of upsetting. And I think, you know, it's very true. It's obviously very true that we've been around for centuries, thousands of years, transgender people, gay people, et cetera, et cetera. It's just that our histories have not, the majority history, the history told by the majority has not allowed those histories to be told, or erased them, or actively erased them. So for what Dr. Wong was saying, that maybe these variants that are associated with transgender people that are being found in this particular study is actually related to, what did he call it here? You know, some sort of potential environmental or social conditions in the past where there may have been a more inclusive space for these different types of identities, which then caused these changes within the genetics, which then leave this mark on our genome. I mean, that's history. That's our history right there, potentially, right there in the genome. I think that's like a really fascinating, just beautiful thought that, you know, and I really appreciated what he was also saying, that this that the genetics really are not determining so much of our phenotypes or our exterior identities, right? I think he used the number of like only say 10 to 20% is due to sort of the genotypes. So really, the genetic code then becomes this marker of history. And so I really like that approach that he's talking about, that he does, of this genetic anthropology, I think, as he called it. Yeah, this is really fascinating and sort of may cause me to take a different direction in terms of how I'm thinking about this project and how to represent some of these things. So I think the best thing to do now with the last, say, half an hour or so of my streaming is to maybe, take some notes on this, maybe type up some things, because I really think I need to take a night, take the rest of the day to think about this, to sort of think about where to go with this project, because it's really quite thought-provoking and really quite beautiful, and I think can really make this quite an amazing project. So I think maybe from here I will switch over to the desktop view, and we can then begin to explore some of this stuff. So why don't I go back to my notes here? Oh, wait a minute. Well, we can maybe do a little bit of an edit here to this VCF file to begin to maybe think about this a little bit more. Just make some notes in here about this. E equals what shall we do here? Let me actually quit Telegram here so my notifications don't come up on stream. Okay, so let's talk about sort of an idea of TH. Number equals, we could 5 type equals type equals string description. Well, let's just do 1 for now. Let's do description. Let's see here. Histories of trans, people in the genomic analysis. Yes. Okay, cool. And let's say we can have like 5 of this. Okay, so let's just think about this. Okay, so we were yesterday we found some of these variants that were found in the exome database on Nomad. So this particular variant, which is a C to T transition, basically found in one African American male. Okay, interesting. So let's start a new thing here about the variants and what other information we can find about this. Okay, database. Okay, so let's just change this up a little bit. Oops. Yeah. Okay, let me just do a bit of formatting here. Oops. Okay. And obviously there's more variants to look through here, but let's just do this for now. Okay, so ctNNA2 has its, let's see here. Oops, oh, there we go. One example, and African / African American male. Okay. Let's go to the next one. So, DIAF2. Oh, there's a lot here. All in women. Interesting. Okay. So, let's just maybe, can I copy this? Oh boy. Okay, so maybe this isn't a good way of collecting some of this information, but maybe I can just do sort of screenshots of this. Very curious about this. Okay, new, save this. I don't know what I'm going to do with this, but maybe this will be something that we can save and show as part of the process of doing this. So sessions, day 2, images. Come on, computer. There we go. Which variant is this? DIAF2. Okay, let me do that for the other one again. Do similar sorts of screenshots for all of this. CTNNA2. Oops. So this is CTNNA2. Okay. Next one. We just won't do any more of that stuff, but we can maybe include those images. Yeah, maybe that's what we'll do. This document that I'm creating of all of my notes, so that we have it later. Hello, come on, load up the variant, please. Oh, there it went. Okay. While that's loading, I can maybe go over here and do something like, I think I can do this in Markdown. Oops. I can do this, I hope. This, and then maybe this. Then maybe images slash, get the right folder here. Projects, missions, events, sessions, day 02, images. Yeah, oops, so that's day 002 slash images. Day 002 slash images slash ctnna2.png. See if I can actually make this. Pandoc, stream notes. I'll put stream notes.pdf. PDF engine equals LaTeX. Template is European. All right, let's see if this works. Okay. Then we can do the same thing here. Hopefully, hope this works. We'll find out soon enough. Day 02 slash images slash IF2 PNG. Cool. Still building. Let's go here. Oops. It's going to be up here, isn't it? Oh, so finished. The image actually here? Oh, excellent. Excellent. So it is there. Great. Great, great, great. Except for I don't know why I've lost some of that. Okay, there's some formatting stuff here that I'm going to need to do. Okay. But let's just finish this up so it's all done. Let's take a screenshot of this variant information. Oops. Oh. Close the snipping tool. Silly me. I closed the snipping tool. Okay, and this is which gene? INHBE. INHBE.png. Okay, great. Let's go back to the code here. AO2 images INHBE.png. Cool. And then the last one, for now at least. Kia 11,009. Okay, and let's take a snip of this. And this is again Kia 1009 PNG. Yes, yes. Okay. AO2 images Kia 109.png. Let me fix this typo here. Cool. And then let's put something there. Oops. Let's put something there. Actually, all of these things should be in brackets. So let me do that. And then brackets here. Oops. Cool. And then let's do the same here. This is just making things look a little bit better in the document, but I think that's important here. Cool. A little bit more to do here. Cool. Okay, great. Let's build this and see what it looks like. And then I'm going to take a sip of coffee. I took a few sips of coffee. Okay. Let's go back to the notes, and I really would like to type up these notes from Dr. Wong, because, yeah, like I said, I really think this project needs to go in a slightly different direction now as a result of that conversation. So let me type some of this stuff up. Okay, great. We have all of that. That's weird. That did not come up with an image. What is that? Here, 1109. Here, 1109. Okay, maybe I'll figure that out later. Notes. Conversation with Dr. Wong. Title. Fragmentations log. Okay, yes, let me make some of these notes here. A lot of it was also just different types of terminology that I found interesting. So the idea of statistical variance and different types of studies, So genome-wide association study, and then phenotype association study, and then his own research about specific variants in the genetic code that are associated with people from Taiwan versus people from the mainland. And then the concept of genetic anthropology I find quite fascinating, as this idea that you can use the genetic code, not just the mitochondrial DNA, which has been used quite a lot, but the genetic code, which I guess, I've seen other studies that have talked about this, but I guess there was something about the phrase that he used that really was quite salient and interesting to me. So then this idea that the changes in sequencing technology leads to different versions of the reference human genome, that there is error rates sometimes of more than 2 to 3% in sequencing, that the Y chromosome is quite difficult to sequence, that maybe more than 40% of it is wrong. And the issue is that many repeats of sequences lead to issues in sequencing. Okay, less than 1% of the total genome is the exome, coding regions. And so, yeah, I guess his term was not genetic anthropology, but molecular anthropology, which I find even more interesting due to some of my previous work with, and ongoing work actually right now, with respect to hormones and molecular signaling within the body. So then there's the contrast between association studies versus causation studies, which leads to the idea that studies of variations, studies of genetic variations, cannot tell you about causation, which is obviously kind of true, but I think it's an important thing to reiterate that in a sense, these are statistical studies of what's different, but we really don't know why things are different and what that means, especially if we're talking about these things like being transgender or so forth, insofar that we can't say, okay, you know, this variant in this gene that causes this change in the protein necessarily causes being transgender. Rather, it's merely associated with being transgender. And it may actually have nothing to do with the phenotype of being transgender. So then he was talking about that something like 10 to 20% of, say, our phenotypes is due to genotypes, which basically also implies that there is no real genetic determinism. Let's see here. Then there was this really, really fascinating concept of metagenomics. So let me do a little bit of research about that. I just want to also highlight here this idea of molecular anthropology, which I think is super fascinating. Okay, metagenomics. Cool. Let me just quickly take a look at this and see if there's some information about this metagenomics that I can include here. Excuse me. Metagenomics allows for the study of different constituents of the human microbiome, including the virome, enabling evaluation of the impact of multiple associations between host and microbe and between microbe and microbe. Okay, well, this is right up my alley. For some stuff later. So let me just make a note of this. Metagenomics and list of papers on Science Direct. And then there we go. Oops. And then insert. Cool. Let's just say something here that this is sort of, yeah, genomic analysis of a combination of species within a particular environment, including the microbiome or virome. Excellent. And then this really beautiful thought. That's the best way that I could describe it. So actually, let's make this all bolded because this is really key. So, more focus on gene histories, on the environmental conditions that triggered these changes in the genetic code. What could have been different in these societies? Maybe they were more open, more equal, allowed for more types of gender and sexual identities and activities. And then how might those differences have been preserved in the genetic code or in the genetic sequences? Because I don't really like thinking about it as a code. Okay, let's call that good. Great. So let's build that. Cool. Let me take a sip of coffee here. Okay, so we have the notes here. Great. Cool. Cool. Still don't know why this isn't working. Why is it not showing up? Maybe there's some issue here. Maybe let me just try something here. So I would like to have the figure captions. Let me remind myself how to do this. And doc images. Oops. Images. Yeah, so that's the way it should work. Okay. Let me see if that's finished yet. Nope. Let's wait for this to finish. I'm just going to go ahead. Huh. It's really not displaying it. Well, that's interesting. Why is it not displaying it? And doc images. Images. Yeah, so this should work. The text would be used as the images alt text. Okay. Huh. Oh, maybe I need this space between things. Right. So let's delete that. Delete that. Delete this. Delete that. Oops, that's why that's not working. Okay. Oh, my eyes are crossed from too many hours of staring at the screen. Okay, now this should give us a nice thing here. I'm just going to quickly make the sort of final image of the day here with some of this information, some of these notes, so that it can go up in the C-Lab space there. Oops. hopefully I can do this relatively quickly. Let me output this as a Word document so that I can get access to it pretty easily. Cool. Great. Let's go to Illustrator, create new. Nope, this needs to be millimeters. We're going to do it as A1. What's the A1 size? A1 size. Paper size. 841 by 594. 41 by 594. This is for print. Come on, Illustrator. Being a little bit slow here. Okay. Let me open up one of the recent files. Okay, I'm going to have to do this differently. Missions, diagrams. Let's do this. Let's copy all of this. Edit, paste in place. Cool. Let me edit this. There we go. Day 002. Variations. Convo with Dr. Wong. Great. Okay, and then let's lock this. Save this as day 02. And then I kind of want to just take some of this code that I've been working on. Wait a minute. I thought I had edited something here. Oh no, I was in the middle of editing this. Histories of right quality of genetic analysis, considering the noise present in all forms of sequencing. Cool. And yeah, we'll work on editing this and creating this something more tomorrow, but let's just copy that. Let's create a big text file, text box here. Let me just paste this. Oops. There we go. Change this to Deja Vu Sans Mono. Bold. Make it quite large. Let's do something like this. Do something like that. Let's actually make this even bigger. Let's make this, say, 120. No. Yeah, 120 is good. Actually, let's make it even bigger. Great. Oops, this needs to go on another layer. I'm going to have to go in a little bit from the stream. Sorry, but I'm going to see what I can finish up here within the next 5 minutes or so. That's sort of just like a quick representation of some of what I've been working on. Great. Let's change the, oops, wait a minute, what's going on here? Oops, that's not what I wanted to do. Oops. Right, no, I was trying to create another layer. Oops, that's what happened. It's put it like that. Okay, great. So let's move that down. Okay, let's change this color to be something more grayish, gray purplish. Yeah, let's do that. And then let's change the opacity. Let's see here. How do I do this again in Illustrator? Layer opacity. Right, properties. That's where everything is these days. Window, properties, I always forget. Oops. Sorry, let me just do this. Cool. Then let's take some of this stuff from our notes. And I guess the main thing I want to do is copy this and create this on another layer here on top of everything. Let's create a text box. Let's lock this bottom layer. Change this to EXO, regular, change it to be like, I don't know, 36. And then we'll have to do a bit of formatting with this. Oops, come on. There we go. Okay, I need to format the paragraph here. Type paragraph. Oh, is it the character? Oops. Oh, that's why. That got way off. Let's do auto. There we go. Much better. Cool. And let's maybe do something like this. And do how do I do this again? Yeah, there we go. That's what I wanted. Cool. Let's make these the same size. Excellent. Let's change this to regular to bold. Metagenomics, regular to bold. And then this thought here, regular to bold. Great. So this is good. Let's align these top. Cool. And then let's create one more layer here. Almost done for the day. Almost done for the day. Let's add these on here. Hopefully those all got on that layer. Yes, they did. Great. Actually, this layer needs to go below everything. Oops. Let's actually lock that layer as well. Cool. And then let's maybe do a bit of filtering on this. Effect, stylize, distort, pixelate. See how this looks. Sure. Let's change the properties of this. Properties. Also bring down the opacity a bit. All right, I think this is good for today. Cool. So I'm going to save this as a PDF. Well, oops. Let's save this as a PDF. I'm going to go over here. I'm going to go in here, get add images. Okay, then I'm going to commit everything. So Deo to notes and screenshots. Cool. Let's push that. All right, so I think I'm going to stop the stream for now. I will be on again tomorrow at 9 a.m. Central European time and 4 p.m. CST. So thanks for any of you who are watching today. Thanks again to Dr. Wong for his wonderful, very thought-provoking conversation with me today. And great. So let's tune in tomorrow and see where we go with this project. Okay, great. Thanks. Bye-bye.